Okay, so in that mucosa and submucosal layers is where you're going to find the circular folds, and you're going to find the villi that I was talking about. So something you need to understand, inside each of these villi, I've got kind of two things. I've got a capillary network, and dead center here, I've got a lacteal, okay? So you know what capillaries are. By now, you should have gone through 22, so you should know what a lacteal is. When it comes to absorbing nutrients, it is very easy to put amino acids and monosaccharides, even disaccharides, into the watery environment of our blood. But fats don't like it. So when we are ingesting, when we're absorbing fats, we have to kind of hide them, okay, in these transport pods. So you've got this fat molecule, and you've just made something this big into something that big. That is a huge change in size. The thing about the capillaries is they have teeny tiny pinholes in them that will not allow something that big in. So we've got lacteals that have these huge openings that actually allow something that big in. So the lacteals help us to absorb the fats. And in every single one of those villi, I've got a capillary network and I've got a lacteal. So I should have no issue absorbing either. The modifications, as I just said a minute ago, are going to greatly increase the surface area. That ultimately greatly enhances absorption. Now, I know everybody's probably heard about gastric bypass surgery by now. One of the things that some doctors do in gastric bypass is they'll take a little piece of the intestine with that gastric bypass. The problem is that when they do that, they actually cause issues because they're taking that surface area away. It would be like saying, I've got the, 36 football fields. I'm sorry. What was it? 3,600 yards, meters, whichever it was. And I just took, instead of 3,600, now I've got, let's say I have 600. That's not a lot of room to absorb. And people that go through gastric bypass, when their doctor does that to them, ultimately end up starving because their body literally does not have enough small intestine to absorb the necessary nutrients to keep their body going. Okay, so looking at the cell types, just like with the stomach, let's look at the cell types that make up the small intestine. You've got absorptive cells, goblet cells, granular cells, and endocrine cells. So the absorptive cells, obviously, they're used for absorption. So their surface is going to have those microvilli, those little folds. Something else that they do, they actually themselves produce digestive enzymes in addition to absorbing. Remember, one of the things I said was that the lining of the intestine, has its own enzymes that it produces. I just ate my sandwich for lunch, so sorry. The goblet cells. The goblet cells in your intestine are going to be the mucus cells. They're the ones that are producing the mucus to, again, line. Just to reiterate, when it comes down to it, you've got to remember, even the enzymes in the intestine can digest the intestine. So again, just like your stomach, you have to have that layer of mucus to protect the lining of the intestine from getting digested itself. The granular cells, they are an immune type of cell, and the belief is that they protect against invading bacteria in the intestine. And then the endocrine cells. If it's endocrine, it's producing hormones, okay? So the intestinal glands are where the cells for the epithelium of the duodenum are produced, and they migrate from the base of the villi to the surface where they're sloughed off. So just like your skin, I go into the shower and I loofah in the morning, I'm sloughing cells off, and that is what they are designed to do. I keep growing more so that when I slough some off, there's some to replace. In here, I do have populations of cells who basically that's their job. So absorptive cells and goblet cells, the top two, they actually go from the bottom of that culvert, kind of like what you saw in the stomach. You'll see it in just a minute. But they go from down here and migrate up to here, okay? So both the absorptive cells do this and the goblet cells, and they can get sloughed off, and it's not a big deal because there will always be kind of a conveyor belt of replacement. They're like escalators, bringing those cells up to the top. The granular cells and the endocrine cells, however, stay in the bottom. Now, why do they stay in the bottom? Well, if I've got immune cells, do I want them sloughing off and going away? Hi, I've got an army to protect me. Let me shoot them. And no, that doesn't make any sense. Same type of thing. And those endocrine cells are producing the regulatory hormones that we've talked about. So having them just kind of slough off and go away, also not a good idea, especially if it's controlling how much product is coming out of the stomach with its very, very high acidity into my small intestine. I don't want those to go away. I want those to be stable. Now, this is the duodenum. So this is that first section of our small intestine. Now, the jejunum and the ileum. Remember, this is that kind of long middle and then the end right before you get to the large intestine. So both actually do have a similar structure to the duodenum, but as you go, the jejunum and the ileum kind of lose diameter. They get a bit smaller. They also decrease in the thickness of the intestinal wall, the number of circular folds, and the number of villi. So as we continue on, our surface area is actually getting smaller and smaller because it's getting less and less wrinkled. Okay, so the first two sections of our small intestine, the duodenum and the jejunum, they're the ones that are primarily responsible for absorption. Okay, they're the ones that do that. Now, I'm not saying that the ileum doesn't do any, it's just by comparison, it doesn't do as much. In the ileum, though, we also have the Peyer's patches. So the Peyer's patches, remember those lymphatic nodules that we saw in the lymph node, those little circles? If you're in my lab, when we did the lymphatic system, we had those lymphatic nodules in, we had them in the pancreas, we had them in the tonsils, and we had them in the lymph nodes. Well, you've got those same lymphatic nodules in places in your body embedded in other organs. And in this case, they're embedded in the ileum of your small intestine. So you've got numerous lymphatic nodules in the submucosa and mucosa of the ileum, that last section of your small intestine. We actually refer to this as the MALT, okay? Mucosa associated lymphatic tissue. Pretty much it is exactly what you're probably thinking it is. It's a part of our lymphatic system that initiates an immune response if there's an invasion in our small intestine. This is where I have those immune cells that I've been talking about ready to go. Now, once I get to the end of the ileum, the first part of my large intestine is called the cecum. So I've got the ileocecal junction, the ileocecal sphincter, and the ileocecal valve, all with that junction from the ileum to that first part of the large intestine called the cecum. So let's talk about the secretions from our accessory organs, okay? As far as the intestine goes, the intestine is secreting mucus primarily. Again, gotta cover everything up. I'm secreting electrolytes, I'm secreting water to kind of keep everything moving. Now, the other things that are getting in there, the pancreatic enzymes from the pancreas, the liver enzymes coming from the liver, and remember I said those cells make their own enzymes. So these are the intestinal enzymes. The duodenal glands, those culverts that we saw, the intestinal glands and the goblet cells produce huge amounts of mucus. Well, we knew that because we talked about the protection. It helps to lubricate and protect the intestinal wall from the acidic chyme that's coming out of the stomach and the action of those intestinal enzymes. Again, they're not cognizant. They don't know what's a good thing or a bad thing. They just digest whatever they come into contact with. Secretin and cholecystokinin will stimulate the hepatic and pancreatic secretions as well. Remember, hepatic is liver, pancreatic, pancreas. The enzymes of the intestinal mucosa stay bound to the membrane, but because of the surface area characteristics, the enzymes still come into contact with the intestinal contents. I've got disaccharidases. They break down disaccharides. I've got peptidases. They break down peptide bonds. Remember, peptide bonds are the two things that hold two amino acids together. You've got a carboxyl and an amine group that are held together. That's a peptide bond. I'm cutting those. So something else. When we talk about most of the things that are being secreted, they're coming in and kind of getting dumped into the hull or the lumen of our intestine. In this case, let's say that this is the lining of my intestine. The enzymes are actually sitting in the cell membrane of these cells. And as that food kind of gets smeared along, the enzymes are working because they come into contact with it regardless, but they're not just being put into this space here. They're staying attached to the cell that they're attached to. Okay, does that make sense? I hope that makes sense. If you have questions, text me or we can get on life size. Okay, so the movement in the small intestine. We kind of already mentioned this. The segmental contractions. So again, I don't want my food, my chyme to just kind of go through as a chunk. I need it to be kind of smushed around so that those enzymes are actually kind of getting into all the nooks and crannies and getting access to everything. So when I talk about segmental contractions, imagine this is the intestine and the muscles are doing this business. If there's a chunk of food in there and I'm doing this, let's say it's a spoonful of peanut butter, and I start doing this business, it's going to get squished and kind of spread around. That's segmental contraction. That's what that means. I'm actually spreading it out by doing this kind of squeezing in the middle of everything. The peristaltic contraction is about moving something a distance. Okay? So in this case, we're squeezing and then pushing along, squeezing and then pushing along. It's not about mixing now, it's about moving. Okay? Stretch of the smooth muscles, local reflexes, and my parasympathetic nervous system are going to stimulate these types of contractions. Now, once I get to the end of the small intestine at the ileum, remember the first part of the large intestine is the cecum, right? So when I get to the cecum, just like when my stomach got something and it started to stretch, that cecum is going to distend. It's going to stretch out. When it does that, it's going to initiate a reflex, basically to inhibit the peristalsis. So it kind of goes back to coming out of the stomach into the small intestine. It's going to kind of do the same thing. The cecum gets something and goes, okay, wait a minute, small intestine, let me deal with what you just gave me, and then I'll get back to you. Just wait a second. It needs time, just like the small intestine needed time. So the cecum distends, it stretches out, and it initiates a reflex to inhibit the peristalsis, that movement, that pushing movement to make it move distally. And this facilitates digestion as well as absorption. So what do I mean? Well, if the stuff is sitting in the intestine for a longer period of time, it's sitting on those absorptive cells for a longer period of time. And if it's sitting on the absorptive cells, not only does that give you more time to absorb from it, but remember all those enzymes that are along the surface, it's exposing that chyme to all of those enzymes for longer periods of time. It gives that set of enzymes along the lining of the intestine more time to break stuff down and absorb. So even though the cecum is going, wait a minute, you need to wait for me to catch up, it's actually helping the small intestine to absorb better. Okay. The rate of chyme movement from the small intestine to the large is slowed down, and it also helps to prevent backflow. I don't know about you, but I don't want the stuff that's in my large intestine coming back up to my mouth. So having a way to kind of control how fast these things are going basically helps to make sure that it only works in one direction. Okay, so let's talk about the accessory organs of our small intestine. So we have the liver. This is the liver. You've got a right lobe, a left lobe. Let's see, you've got the quadrate lobe. Here's my gallbladder. And yes, believe it or not, it is actually green. It looks kind of like a deep forest green, but it is actually green. So this is the bottom of the liver. This is the anterior view, like you're looking this way. This is as if I flipped it over, and this is looking from the top, like you're looking from where the neck is downward, okay? So you've got the 4 lobes. You've got the right and left lobe, the caudate lobe, and the quadrate lobe. I don't think I pointed out the caudate lobe, so that's this one. And you've got the porta. The porta is going to be on the undersurface. You can see it right here. You've basically got vessels going in and out. You've got ducts, tubes, and nerves that enter and exit the liver here. This is my entrance or exit. No matter what I am, this is where I'm going in or going out, okay? In that porta, you have the hepatic portal vein. Okay, so what's a portal? Again, if you watch sci-fi, you know what a portal is. It's going to be basically a gateway or a doorway that carries things from point A to point B, okay? If you've ever seen Stargate, that's a portal. It took them to another world. The stuff that we have digested, the nutrients that we have digested and taken from my stomach, my intestines, all of that needs to be cleared before it's allowed to go into the rest of the body. I need to make sure that if something is dangerous, it gets broken down. I need to make sure that if something didn't quite get digested as well as it should, that it finishes and gets, digested the right way. The portal system is actually taking all of the stuff that we've absorbed from the stomach, the intestine, and bringing it to the liver so the liver can check everything. That's the portal. The hepatic portal vein takes stuff from my digestive system over to the liver, basically for double check, okay? So that hepatic portal vein takes the blood from the stomach, the small intestine, the large intestine to the liver, okay? The hepatic artery right here, and it's in red, notice. This is just like any other artery. Arteries carry blood away from the heart. This is carrying freshly oxygenated blood to the liver cells. That's it. The common hepatic duct is where the left and right hepatic duct meet. So one of the things that your liver does is it makes something called bile. Bile is one of the things that helps us to break down fat. It helps us to emulsify fat. So I've got the left and right lobes. They each have one major tube that collects the bile and brings it down. And those are going to be the common hepatic ducts, which you can see here, okay, in green. Now, the cystic duct. Remember I said hepatic always means liver. Cystic always means gallbladder, okay, when it comes to A&P anyway. So the cystic duct is a tube coming out of the gallbladder carrying bile as well. So it'll also be green in any picture. Now the common bile duct. It carries bile from the liver and the gallbladder to the duodenum, to the first part of my small intestine. So I've got a major duct coming from the liver. I've got a major duct coming from the gallbladder, and they combine into one big duct, okay? That one big duct is going to be the common bile duct. Why? Because they're both carrying bile. They're both carrying the same thing. And this will look a lot better in the next slide. Now, the hepatopancreatic ampulla. I was going to say ampulla. I was like, what? This is where the duct from the pancreas, the tube from the pancreas, and the common bile duct fuse together. Okay, so let me show you. All right. So going from the underside, I have this hepatic portal vein that's bringing all of the stuff that I've absorbed from the stomach, the small intestine, large intestine up to the liver so that it can be processed. I've got the hepatic artery, okay, that's coming in, basically bringing freshly oxygenated blood. Now here are my two lobes, right? Correct? I've got my right lobe and my left lobe. There are the two ducts carrying bile, the left and the right hepatic ducts. They fuse together here into the common hepatic duct. Here's my gallbladder. There's the cystic duct. Remember, duct is just a fancy way of saying tube. So this is carrying bile, it's green. These are carrying bile, green. They fuse to make this, bile. And then these two come together. This is that common bile duct, bringing bile from my gallbladder and from my liver, okay? As it comes down, it fuses with this duct. Here's my pancreas, right? This is the pancreatic duct. When they fuse, it becomes the hepatopancreatic ampulla. Hepato, liver, pancreatic, pancreas, ampulla, tube, okay? So, the only thing that is, as far as the cystic duct, the left and right hepatic ducts, the common hepatic duct and the common bile duct, all those are carrying bile, period. That's all they're carrying. The pancreatic duct carries pancreatic juice. I know it sounds gross, but that's what it's called. When they get to the hepatopancreatic ampulla, it's going to be a mixture of those two things before it gets into the small intestine.